Hey guys, welcome back. Today I am just going to rehash what we did yesterday. It is Saturday 4/11. If you missed our live video session, our Zoom session yesterday, you're going to get it all again right here and you can watch it as many times as you'd like. We covered three main topics in the digestive system. The first topic of conversation are the four tunics or layers of tissue of the alimentary canal. The second topic that we covered were its microanatomy of the stomach. We did the four cell types in a gastric gland and descriptions of those cells, what they do. And finally, the third topic, the most complicated, we talked about the three phases of gastric secretion. So I'm going to go over that all in depth right now. So topic number one, the four tunics or layers of the alimentary canal just refers to the wall of that tube. Our digestive system is basically just a long tube from the mouth all the way down to the anus. And that tube is made up of thickness of the wall of the tube has several layers of tissue associated with it. It's generally the same, at least the layers are identified as the same four layers from the mouth all the way down to the anus. The four layers of tissue, what's in them is generally the same from the lower esophagus all the way down through the large intestine with some variation. The two ends, the mouth and the anus, are a little bit different. So we'll start right off the bat. The inside layer that lines the tube, we call layer one or tunic number one, that's called the mucosa. The reason it's called the mucosa is because it secretes a lot of mucus. So I want to make sure that I cover everything in my notes for you. So layer number one is called the mucosa. The tissue on the lining on the top layer of the mucosa that's actually pointing toward the lumen of the alimentary canal where the food is, that's generally made up of simple columnar epithelial cells. But that's generally from the lower esophagus down through the large intestine. The lining, that top layer in the mouth, pharynx, and upper esophagus, as well as the anus, is stratified squamous epithelium. So the mucosa layer is either simple columnar epithelial cells or stratified squamous epithelial cells depending on the location. Now the reason that the mouth, pharynx, upper esophagus, and anus that are lined with stratified squamous epithelial cells is for protection. Those are areas where we get abrasion, they're exposed to the outside world. So that stratified squamous layer of epithelium is for protection. The rest of it, from the lower esophagus all the way down through the large intestine, is simple columnar epithelium. And those cells generally function in secretion, and they secrete mucus, enzymes, and hormones. So function number one of the mucosa, other than the mouth, pharynx, upper esophagus, and anus, the function is secretion of mucus, enzymes, and hormones. The second function is absorption. So in certain places along the alimentary canal, we absorb things. We absorb nutrition, nutrients. The stomach absorbs very few things, a couple, but very few. The majority of our absorption goes on in the small intestine. That's where most of our nutrition is absorbed from our food. And finally, in the large intestine, it's mostly just reabsorption of water that goes on there. So we've got secretion, absorption, and finally there is a protective function of that mucosal lining because it does provide a little bit of a barrier for things crossing into our bloodstream. So that's the mucosa. It's made up of simple columnar epithelium mostly. There's also a lamina propria just deep to that. That's basically connective tissue, a little bit of areolar connective tissue, some immune vessels, some lymphatic vessels. And then deep to that is a really thin ribbon layer of muscle. This is all still in the mucosa layer. So there's simple columnar epithelial cells, lamina propria, and a thin layer ribbon of smooth muscle called the muscularis mucosa. That function of that little ribbon of smooth muscle is local motility. Provides some motility, but just generally in a local spot, one location. The second layer, So we've done the mucosal layer. Deep to the mucosa is a layer called the submucosa. Sub means below, mucosa. So the submucosa is generally made up of areolar connective tissue. Its characteristic feature is it's highly vascularized and also very well innervated. So lots of nerves, lots of blood vessels. Whenever I hear submucosa in the GI tract, I think nutrition. That's its main function. It supplies nutrition to the tissues all around it, both the mucosal layer and a bit the submucosa, of course, as well as the muscularis externa below it. So the main function of that submucosal layer is nutrition, highly vascularized and innervated. The third layer, deep to the submucosa, is the muscularis externa. It's generally very thick, not in all places, but especially in the esophagus, stomach, some parts of the large intestine, and the large intestine. This muscularis externa layer is extremely thick relative to the other layers. It's generally made up of two layers of smooth muscle, a circular layer and a longitudinal layer. That just refers to the direction of the fibers of the muscle cells. So if we have a tube, the digestive system, the alimentary canal is just a tube. The circular layer of smooth muscle just means the fibers are running this way, sort of like a coiled spring. The longitudinal fibers are running parallel with the direction of the tube. Main function of the muscularis externa is motility in general, specifically peristalsis and segmentation. Peristalsis means squeezing that movement of food material along that tube from one location to another. Segmentation generally means the churning of contents in one place, sort of like a washing machine. So we've done mucosa, submucosa, muscularis externa, and the fourth and final layer or tunic of the alimentary canal is called the serosa. The serosa is synonymous in the GI tract with the visceral peritoneum. So that means the outer covering of the organs, that sort of clear, shiny connective tissue that covers our stomach, our intestines, the esophagus. That's called the serosa. It's generally coarse connective tissue. It's tissue makeup, and its function is basically structure and protection of those organs. All right, that's topic one, the four tunics of the alimentary canal. Now we're going to go to microanatomy of the stomach. And this is headed under the stomach in general. Microanatomy, we're going to look at the cell types in a gastric gland. So before we do that, I'm going to draw on the board behind me a very simple gastric gland so that we can label these cells. This represents the wall of the stomach. That means tissue. So this is all solid tissue. This is the lumen of the stomach up here. That's where we find food content. So this is the space inside the stomach. This is the wall of the stomach. This structure represents, this sort of laboratory bottle-shaped structure represents a gastric gland. And it would be a simple gland in its structural arrangement. Generally, the secretory cells of a gland are down here in this part, and then the gland duct rises up to the lumen where the secretions empty. Most glands, including a gastric gland, are basically loaded with cuboidal cells. So I'm just drawing these box cube-shaped structures that represents the secretory cells of the gland. Now I'm just going to go through the cell types and their secretions and their locations. So the first cell type on this list is called a mucous neck cell. Mucus because it secretes mucus. Neck cell because it's located up here almost in the duct of the gland. So I'm going to color in a couple of cells here, and we're going to label those M N cells for mucous neck cells. They secrete sort of a thin acidic mucus, and the function is not really that well known. That thin acidic mucus is secreted into the duct and then upward into the lumen of the stomach. Second cell type we're going to talk about is a parietal cell. So I'm going to color in 2 more cells here, and I'm just going to label it a P for parietal. The parietal cells are generally located in what's called the apical region of the gland. That's up here where it starts to constrict, so sort of at the shoulder of the gland. So we have mucous neck cells, they're almost in the same place. Mucous neck cells, parietal cells, and those parietal cells basically do 2 major functions. The first is the secretion of hydrochloric acid. So that's something you need to tattoo on your brain. Parietal cell secretes hydrochloric acid. That acid is secreted here and then makes its way up into the lumen, where that hydrochloric acid breaks down food, predominantly proteins. The second function, main function of a parietal cell, is that it secretes intrinsic factor, which is a hormone molecule. So HCl, hydrochloric acid, and intrinsic factor, both secreted by a parietal cell. Intrinsic factor is a life-sustaining function of the stomach, meaning we need that in order to survive. Intrinsic factor is necessary for the proper absorption of vitamin B12 in our diet. Without vitamin B12 absorption, our red blood cells don't develop normally, so it is required to sustain life. So if you're ever asked what's the main life-sustaining function of the stomach, that's it, the secretion of intrinsic factor. The third type of cell is called a chief cell. Those are located further down, getting toward the base of the gland. So there's a chief cell on both sides of this gland. And chief cells generally secrete digestive enzymes. One big one in particular is pepsinogen. Pepsinogen is the inactive form of the enzyme pepsin. And the enzyme pepsin breaks down proteins. It's pepsin from peptide. It breaks down proteins into peptide fragments. So it denatures proteins. It helps digest proteins. So HCl and pepsin break down proteins when they hit the stomach. Now, chief cells don't secrete pepsin right off the bat. They secrete an inactive form of that enzyme called pepsinogen. I'm going to write that over here. So a chief cell secretes pepsinogen, which is inactive. And when that inactive enzyme is secreted, makes its way up here, and it combines with hydrochloric acid from a parietal cell up here, then it becomes active and can do its digesting. So pepsinogen plus HCl gives us pepsin, which is active. So chief cells first, they secrete an inactive form of pepsin called pepsinogen. When it combines with hydrochloric acid, it becomes active and can do its digesting. The reason that's a good thing, or why this occurs, is we don't want an active, highly active enzyme being secreted right here, or it will start to digest the wall of the gland itself. So it's inactive, makes its way all the way up here. When it combines with HCl, then it becomes active to start digesting the food. Chief cells, another main function besides secretion of pepsinogen is the secretion of lipases. Lipase, lipase, that's ACE just means enzyme, LIP is for lipid, so it's an enzyme that breaks down lipids. So we've done mucous neck cells, parietal cells, chief cells, and the last, it's actually a small group of cells, is called enteroendocrine cells. And those secrete hormone-like substances. It's a whole group of cells, enteroendocrine cells. Of those cells, I'm going to give you a short list of the substances secreted, and we're going to look at one of those cells in particular and what it secretes. So I'm going to move my P for parietal a little closer to the gland. And in this space here, I'm just going to write the main components. They're hormone-like structures. They're either paracrines or considered real hormones. I'm going to list what is secreted by enteroendocrine cells. So I'm going to write EE, enteroendocrine cells, and they secrete histamine, serotonin, somatostatin, and I'm going to put a star next to this last one, gastrin. Gastrin is a hormone that's secreted by G cells. That's where they get their name. I'm going to put from G cells. And those are located, actually most of these enteroendocrine cells are located down here in the very base of the gland. So there's our G cell. And the main function of a G cell is to secrete the hormone gastrin. It's technically a paracrine that acts on a neighboring cell. So here's the chain of events. When a G cell secretes the paracrine or hormone gastrin, that paracrine, that component, that molecule, binds to a parietal cell and enhances the secretion of hydrochloric acid, sort of turns it on, if you will. So whatever stimulates a G cell to release gastrin, that gastrin then binds to a parietal cell and enhances the secretion of hydrochloric acid up into here. So those are the four main cells. The last one was really a group of cells, enteroendocrine cells, specifically a G cell is one type of enteroendocrine cell. So we've done mucous neck cells, parietal cells, chief cells, enteroendocrine cells, specifically a G cell. That takes care of the second topic of this lecture. The last one is the three phases of gastric secretion. So I'm going to erase all of this business. I'm going to list the three main phases of gastric secretion, talk about each one in detail, and then we can call it a day, wrap it up. So, three phases of gastric secretion. That just means what causes the stomach to start secreting things and digesting. The first phase, number one, is called the cephalic phase. Cephalic means head. The reason it gets that name is because this is sort of psychological. This phase occurs before food ever even enters the stomach. And it can be triggered by the thought of food, the smell of food, the taste of food. It can even be triggered by the time of day. For example, if you eat dinner every day at 6 p.m. and 6:30 rolls around, your stomach starts growling and it starts secreting because it's timed, it's conditioned, the stomach is conditioned to start secreting and digesting things at about 6:30. So this happens before food enters the stomach. It is a psychological conditioned response. And what sends signals from the brain, it's the medulla oblongata, what sends signals is via the vagus nerve to the stomach from the medulla. That's the easy one, cephalic phase. The second phase is called the gastric phase. I know that seems redundant. We're talking about gastric secretions, and this is the gastric phase. But gastric just means stomach. Cephalic means head. So this occurs from the brain before food enters the stomach. This phase, second phase, starts occurring when food actually enters the stomach. That's why it's called the gastric phase. there's two main components to this phase. First of all, this is responsible for most of gastric secretions, about 60%, roughly 2/3 of gastric secretions come from phase 2 after food actually enters the stomach. And there's two main stimulatory factors or mechanisms by which this works. There's a mechanical one and there's a chemical one. So there's mechanical stimulus and chemical stimulus. I'm going to talk about the first one. It's very easy. It just means when food enters the stomach, there are stretch receptors in the wall of the stomach that when food enters and the volume increases, it stretches the wall of the stomach, expands. There are stretch receptors in the wall of the stomach that sense that stretching. And they send a signal to the medulla, and then the medulla says, there's food in the stomach, sends a signal back via the vagus nerve, tells the stomach to start secreting things. So that's number one. Distension of the stomach activates stretch receptors. The second stimulatory factor is really chemical stimulus or chemically activated, and it's from a change in pH mostly. When proteins enter the stomach, which I'm going to talk about that just for about 30 seconds. Digestion begins as soon as food hits our mouth. So we chew up food, that's considered a digestive mechanism. There are enzymes in our saliva that begin the breakdown of things like sugars, carbohydrates, that's amylase, and lipases are also secreted in saliva to break down lipids. However, there are no enzymes in our saliva to start breaking down proteins. So proteins pretty much stay intact until they hit the stomach. That's where protein digestion occurs. It begins, and most of it occurs. So in the gastric phase, we're talking about the second stimulus of, we talked about mechanical receptors, now I'm talking about a change in chemistry or the chemical environment of the stomach. When proteins hit the stomach, pH rises, it becomes more alkaline. That stimulates the stomach to secrete more hydrochloric acid in particular. So proteins hit the stomach, pH goes up, G cells secrete gastrin, gastrin binds to a parietal cell and tells a parietal cell to secrete hydrochloric acid, initiates that. Hydrochloric acid then is secreted into the stomach and starts to break down denatured proteins. That's the chemical stimulus of it. It's sort of the chemical part is also sort of a negative feedback mechanism. It's self-regulating. When proteins hit the stomach, pH goes up, gastrin secreted, hydrochloric acid is secreted, proteins start to get denatured or broken apart. Then as they get broken down, pH starts to return to normal, starts to decrease, and then gastrin secretion decreases, hydrochloric acid secretion decreases. It's sort of a self-inhibiting effect after the pH drops. Now the third and final phase of gastric secretion is called the intestinal phase. Phase 1, before food enters the stomach. Phase 2, when food actually enters the stomach. Phase 3, this is when chyme from the stomach or digested foodstuff from the stomach enters the intestines, specifically the duodenum, the first section of the intestine. So this phase is what happens when chyme is released from the stomach into the duodenum via that pyloric sphincter. Now in the intestinal phase, there's a couple simple things to learn right off the bat to keep in mind. First, in the intestinal phase, the first part of it is stimulatory. The second part of the intestinal phase is inhibitory. So first, when chyme enters the duodenum, there are signals sent back to the stomach to stimulate more gastric secretion. It's stimulatory. It enhances stomach secretions. But that's very short-lived, and then it quickly it turns into an inhibitory response. Theory is something like this. Chyme enters the duodenum. When it does, the sensors, and I'll talk about those specifically, feed back to the stomach and say, increase your secretions. Basically, it's the intestine telling the stomach, finish digestion. Secrete, hurry up, secrete more acid, get digestion done, finish. But that is very short-lived, and then that flips and turns into an inhibitory response. The intestine tells the stomach, all right, shut off, shut down, quit secreting. If you don't quit secreting and hydrochloric acid keeps emptying into the duodenum, you're going to fry the wall of the duodenum. So there's a turn-off mechanism that's second to the enhancement. Or all right, so these are the specifics of the intestinal phase. We know the first one is stimulatory, the second action is inhibitory. Now, during the stimulatory phase, when food or chyme enters the duodenum, it stimulates the intestinal mucosa cells to release something called intestinal enteric gastrin. So it's a form of gastrin coming from the duodenum that feeds back onto the parietal cells of the stomach, tells them to enhance the secretion of hydrochloric acid. That's the stimulatory phase. That's very short-lived. Then it flips over to inhibition, or an inhibitory phase. And what happens during the inhibitory phase is it's caused by, first, duodenal distension, meaning stuff, chyme entering the duodenum causes the wall to stretch. There are stretch receptors. Also, second, the presence of acids in the duodenum helps cause the inhibition back on the stomach. And third, the dumping of fats from the stomach, lipids from the stomach into the duodenum, also helps shut down gastric secretion. What we want to do is prevent excess acidity from getting into the duodenum. So it's telling the stomach to shut things down. Here are the mechanisms now of inhibition. First is called the enterogastric reflex. So I'm just going to write mechanisms of inhibition. Remember, that occurs after the stimulatory mechanisms are over. So the mechanisms of inhibition are first what's called an enterogastric reflex. During the enterogastric reflex, this is a neural response. There are short reflexes of the enteric nervous system. That just means they're local. The nerve fibers are extremely short, and they're just connected from the duodenum to the stomach and vice versa. So we have short reflexes of enteric nervous system, and there's also a long reflex going all the way to the medulla and then back to the stomach via the vagus nerve. So there's a short reflex, long reflex that inhibit acid secretion in the stomach. So I'm going to write neural, colon, short and long reflex. The second mechanism of this enterogastric reflex is called enterogastrones, means hormones of the intestine. Enterogastrones are released. There are enteroendocrine cells in the mucosa of the duodenum that release a group of hormones called enterogastrones. The two main ones that act back on the stomach are secretin, secretin, and CCK, or cholecystokinin. Those two hormones feed back, they inhibit gastric secretion. They also inhibit other digestive functions, but mainly gastric secretions coming from the stomach. So there you have it. Those are the three major phases of gastric secretion and how they work. Next lecture, I'm going to talk about actually how a parietal cell secretes, the mechanisms involved for the secretion of that hydrochloric acid when, for example, gastrin binds to the plasma membrane of that cell.